Hi, I'm Tyler Vernon, and today we're going to be discussing how the differentiation of human airway epithelia affects ACE2 receptor expression and infection of severe acute respiratory syndrome. The study ACE2 receptor expression and a severe acute respiratory syndrome coronavirus infection depends on differentiation of human airway epithelia was conducted by Jia Hongping et al. In this study, researchers aimed to assess where the ACE2 protein was more abundantly expressed by using immunofluorescence staining and surface biotinylation. First, they tested whether the ACE2 receptor appeared more on the apical or basolateral surface by immunohistochemistry. Then, they determined if cell differentiation made a difference in the expression of ACE2 by using a scanning electron microscope to compare the surface morphology of differentiated epithelia. The results of the study support the notion that the infection of airway epithelia tissue by SARS-CoV in humans is directly influenced by the levels of ACE2 mRNA expression and cell differentiation. This paper highlighted three major characteristics of epithelial cells that influence infectivity. First, SARS-CoV preferentially infects cells with ciliated membranes due to elevated levels of ACE2 expression. Image 1 shows positive correlation between ciliation and ACE2 expression. Image 2 then continues to show the positive correlation between ciliated cells and SARS-CoV infection. Second, SARS-CoV preferentially infects cells from the apical surface rather than the basolateral surface. In image 3, the presence of SARS-CoV is shown through the presence of the SARS-CoV NSP1 replicase protein illuminated in green. Finally, SARS-CoV preferentially leaves the cells through the apical surface of cells rather than the basolateral surface. In image 4, levels of SARS-CoV regression were measured and were significantly higher on the apical surface. This research study connects with our study in molecular cell biology since we have learned the cell differentiation pathways as well as receptor protein functions. We have learned that in order for a virus to make entry into a cell, a receptor protein is needed. These receptor proteins are embedded in the cell membrane and allow for molecules to affect cell function without actually entering the cell. Finding the way to block the virus from binding to the ACE2 receptor is crucial in order to prevent infection. This study is important because it shows how SARS-CoV affects the human airway, which can be crucial in identifying the susceptibility of infection. Identifying what makes a person more susceptible can help with research on developing a therapy for coronavirus, such as using antibodies to block the ACE2 receptors. The research also helps to identify how the viral infection can get into its host, and therefore how we can prevent it by taking part in things such as social distancing and frequently washing our hands.